Good day, everyone. I'm Tricia Maylani Marmo.
And I am Mary Amethyst Tome.
Today, we will be presenting our study on the Novo design of antimicrobial peptides, AMPs using computational and experimental approaches. This research aims to address the growing challenge of antimicrobial resistance by developing innovative peptide-based therapeutics.
Antimicrobial resistance, or AMR, is a major global health issue. Conventional antibiotics are losing their effectiveness due to bacterial resistance, which makes the search for alternative treatments crucial. Antimicrobial peptides, or AMPs, are promising candidates because of their broad spectrum activity and unique mechanisms that reduce the likelihood of resistance. However, naturally occurring AMPs often have limitations, such as toxicity or instability. This study focuses on designing novel AMPs using de novo computational tools, optimizing their properties to enhance antimicrobial activity while ensuring safety.
The problem we aim to address is the increasing resistance to current antibiotics. While AMPs offer potential solutions, challenges remain, including high toxicity to human cells, limited stability in physiological conditions, and narrow spectrum activity. Our goal is to design and optimize new AMPs that overcome these challenges.
To tackle these challenges, our study has the following objectives. First is to design novel AMPs using computational tools and AI models. Second, evaluate their physicochemical properties to ensure antimicrobial potential. Third, assess peptide stability using molecular dynamics simulations. Fourth, synthesize and characterize the most promising AMPs. Fifth, test their antimicrobial activity using against gram-positive and gram-negative bacteria. Last is to evaluate toxicity on human red blood cells and mammalian cell lines. By achieving these objectives, we hope to contribute to the development of safer and more effective antimicrobial agents.
This research has significant implications across various fields. First, scientific community. Advances our understanding of computational peptide design and antimicrobial mechanisms. Second is the in the medical field, provides potential candidates for future antibiotics. Lastly is for biotechnology industry, contributes to the drug discovery and pharmaceutical advancements.
Previous studies have demonstrated the potential of AI-driven AMP design. In a study of Lin et al., they used a deep learning model called Wasserstein-Vian with gradient penalty or vegan GP to generate AMPs with optimized antimicrobial properties. Their findings showed that AI can effectively accelerate AMP discovery. While in the study of Fresser et al., they focused on computational modeling to design AMPs with enhanced potency and lower toxicity. They validated their results through molecular dynamic simulations and peptide synthesis.
While previous research has provided valuable insights into antimicrobial peptide design, there are still some important gaps and limitations. For the first study, it relied heavily on in silico predictions and validated only at 8 peptides experimentally. This limited experimental validation may restrict the generalizability of their findings and the practical application of the designed peptides. For the second study, it focused on structural optimizations of AMPs but did not incorporate high throughput generation methods. This limitation could slow down the discovery process, making it harder to identify large numbers of promising peptides quickly. These gaps highlight the need for a more comprehensive, high throughput approach that combines computational predictions with robust experimental validation across a broader range of peptides and bacterial strains.
Our study is guided by three key frameworks. First is the AI and deep learning in drug design. We use GAN-based models to generate optimized peptide sequences. Second is the protein structure and biophysics. Computational screening ensures that designed peptides remain stable and selectively target bacterial membranes. Third is the experimental validation, using techniques such as solid phase peptide synthesis, antimicrobial assays, and cytotoxicity studies to confirm theoretical predictions.
Our study follows an in silico experimental hybrid approach to design and validate novel antimicrobial peptides. First, data collection. Antimicrobial peptide sequences will be gathered from public databases with peptides shorter than 10 amino acids or longer than 30 amino acids, excluded to maintain structural relevance. Second one is the peptide design and generation. We will use the PC6 encoding method to represent peptides as numerical matrices. A buzzer's time GAN with gradient penalty or WGAN GP will generate novel AMP sequences, ensuring diversity and optimization. Third, in silico screening, the antimicrobial potential of the generated peptides will be predicted using AI4 AMP. Candidates will be selected based on physical chemical properties and sequence uniqueness to maximize potency while minimizing toxicity. Fourth, structural and functional analysis. Molecular dynamic simulations will be conducted to assess peptide stability and membrane interactions, ensuring they remain effective in physiological conditions. Lastly is the experimental validation. Selected peptides will be synthesized via solid phase peptide synthesis and tested for antimicrobial activity using distant fusion and minimum inhibitory concentration assays. Safety will be assessed through hemolysis and cytotoxicity assays to ensure they do not harm human cells. Through this comprehensive methodology, we aim to design effective, safe, and stable antimicrobial peptides, bridging computational predictions with real-world applications.
As for the recommendations we gathered from the literatures that we collected, Lin et al. Tested only 8 peptides, which limits applicability. They recommend to increase experimental validation. Future research should validate a larger set using in vitro and in vivo studies. Additionally, high throughput screening should be incorporated. Fraser et al. Optimized structures computationally but lacked rapid generation methods, which could slow down the discovery process.
This concludes our presentation. Thank you for your time, and we are happy to take any questions.